Hey everyone, and welcome to this explainer. I'm super excited to guide you through today's topic. We're zooming right in on the very first true embryology moment in the IVF laboratory workflow. If you've ever found yourself wondering what actually happens the exact second an egg is retrieved from a patient, well, you're in the right place. It is a fascinating, high-stakes journey of precision and care. So let's just set the scene and follow the science right from the follicle to the dish. Okay, let's kick things off by busting a pretty massive common misconception right out of the gate. In the IVF world, what arrives in the lab is possibility, not a visible egg count. The moment those eggs are collected by the doctor is definitely not the moment they're magically ready for fertilization. In fact, the work is really just getting started. Somebody still has to actually find them, identify them, protect them, and prepare them. So the crucial thing we need to understand here is what's actually inside that test tube being handed over to the lab. We've got to completely scrap this myth that doctors are just handing over a neat, tidy little tray of visible eggs. That's not how it works. Instead, the lab receives what we call a follicular aspirate. Think of this as a highly complex, frankly messy, biological soup. It contains follicular fluid, blood, granulosa cells, cellular debris, and, if we're lucky, a cumulus oocyte complex hidden deep, deep inside all of that material. It is a raw environment, and the minute that tube is handed over, the clock is ticking. To make sense of all this, we're going to hit 5 main checkpoints today: the critical lab handoff, searching the fluid, recovering and washing the eggs, the difference between counting and biological maturity, and finally, witnessing and identity control. Let's see exactly how this chronological journey unfolds. Starting right at checkpoint 1, the critical lab handoff. It's a race, but definitely not a rush. You see, there is a very distinct line between the clinical procedure and the lab workflow. The clinical procedure is what the doctor is doing, which is aspirating the follicles under ultrasound guidance. But the highly specialized specimen recovery, that is performed entirely by the embryologist. The literal second the aspirate leaves the patient, the embryologist takes the baton. They immediately initiate temperature control, start an ultra-efficient search, and lock down strict identity protection. So while egg collection and egg recovery are totally linked, they are completely different events, requiring vastly different skill sets. Which brings us to step 2, searching the follicular fluid, basically finding the needle in the haystack. Now, you might be wondering, what exactly are they looking for? You might picture them scanning this bloody, messy fluid for tiny, bare eggs just floating out in the open. But what are they genuinely trying to spot under that stereo microscope? Well, I can tell you right now, it is absolutely not a bare egg. The embryologist is actually hunting for a soft, fluffy cloud of cells known as the cumulus oocyte complex, or COC for short, because the oocyte itself is hidden deep inside its outer shell, which is called the zona pellucida, and then it's completely surrounded by all these cumulus and corona cells, so the whole structure just ends up looking like a soft, cloud-like mass floating in the debris. The embryologist relies on some intensely trained visual discipline to spot these tiny, delicate little clouds amidst a literal sea of red blood cells and tissue. It requires some serious focus. Once they actually spot that cloud, we enter phase 3, recovering and washing the oocytes. This is all about incredibly safe handling. This recovery phase builds into this really beautiful, seamless flow. First, they methodically scan the fluid. Then, they gently pick up that delicate COC using a very specialized pipette. And finally, they transfer the complex right into clean, prepared culture media. This crucial step washes away the contaminating blood and debris, basically gently rescuing the complex from that harsh environment and placing it into a pristine holding dish. It's essentially a delicate rescue operation happening entirely on a microscopic scale. But, technique isn't the only thing that matters here. Throughout this entire process, there is one highly specific number dictating every single move: 37.0 degrees Celsius. Temperature stability is absolutely paramount because oocytes are incredibly sensitive to their environment. Even the slightest drop in temperature can harm the structural integrity of the egg, potentially damaging the really delicate internal mechanisms it needs for fertilization and future embryo development. A temperature drop is basically a biological shock, and the lab absolutely must avoid it at all costs. So the embryologist has to maintain unwavering environmental control. Things like pre-warmed media, a perfectly stable pH, and absolutely minimizing the time the egg spends outside those controlled incubators are totally non-negotiable rules for oocyte survival. The embryologist is constantly doing this balancing act, incredible speed mixed with absolute delicate precision, so these single cells aren't stressed for even a fraction of a second. It's a highly choreographed routine where every single movement is optimized. Okay, as the dishes are safely organized, let's talk about our fourth checkpoint, counting versus biological maturity. What do these numbers actually mean?
It's really time we redefined success here. When a patient hears the number of eggs recovered, that metric simply means the total number of cumulus oocyte complexes the lab found in the fluid at the end of the ovum pickup. But mature eggs, meaning the ones that are biologically ready to actually be fertilized to creating embryo, that metric is assessed much later on. We only know that after the protective cloud of cells is chemically and mechanically removed in a process called denudation. So recovery is literally just the beginning of the evaluation. It is not the final verdict on maturity.
Let's pause for a quick reality check, because we really need to bust the myth that a follicle count seen on an ultrasound strictly equals your final egg count. It just doesn't. Not every follicle contains an egg, and even if you do have a high follicle count, it doesn't guarantee a simple or high-yield recovery in the lab. The embryologist might run into tricky granular fluid or excessive blood that makes finding the eggs super technically demanding. The ultimate success of the recovery depends heavily on the unique biology of that specific aspirate on that specific day. All right, let's zoom out from the microscope for our fifth and final checkpoint, witnessing and identity control. Above all else, this is about protecting the patient. Think about it. You could have the absolute most perfect biological handling in the world, but a flawlessly handled oocyte is totally useless if identity control is ever compromised. That makes rigorous traceability just as critical as the biological handling itself. Every single tube, dish, and transfer point must perfectly match the patient's identity. Counts are meticulously documented in real time. Both manual and electronic witnessing systems actively lock in that identity at every single transfer step. And there is continuous, open communication flowing back and forth with the clinical team. It is a strict, zero-tolerance environment for errors. You can truly appreciate the incredible choreography of this whole process when you just break down the minutes. It starts right at minute 0 with the handoff, moves immediately into the stereo microscope search for the first couple of minutes, transitions straight into gentle washing and transferring, and by minute 5, those cells are fully secured in their incubator. The stakes are so high that every single second is accounted for. Seeing it mapped out chronologically like this beautifully illustrates exactly why the initial lab recovery is heavily disciplined and always described as a race, but never ever a rush. So, the recovery is complete. The identity is locked in. The oocytes are resting safely and comfortably at exactly 37 degrees. But what biological secrets are these oocytes going to reveal when their protective clouds are finally removed? Every single embryology decision moving forward depends entirely on what was successfully recovered and protected at this crucial first stage, leaving us to wonder what we'll discover when we assess the true maturity of these oocytes in the very next phase. I really hope this explainer has given you a profound new appreciation for the incredible science happening every day inside the IVF lab. Keep learning, stay curious, and I'll catch you next time.